In this podcast, we'll look at sperm production. Now, spermatogenesis is the process by which spermatogonia develop into sperm. Spermatogenesis begins shortly before puberty under rising levels of gonadotrophin, and spermatogenesis continues for the majority of a man's life, can actually occur up until the 8th decade, sometimes even longer. Spermatogenesis is a process that involves both mitosis and meiosis. The spermatogonial phase, in this phase spermatogonia divide by mitosis to replace themselves and to provide a population of committed cells that will undergo meiosis. Basically, in the spermatogonial phase, cells are going from 2n2c to 2n4c and then back down to 2n2c. The spermatocyte phase is where spermatocytes undergo meiosis to reduce chromosome number and DNA content, producing haploid spermatids. So the cells go from 2n2c to 2n4c before the first meiotic division. At the end of the first meiotic division, the cells are 1n2c. At the end of the second meiotic division, the cells are 1n1c. Now, I want to point out that secondary spermatocytes don't stay around for that long before they start becoming spermatids, so histologically it's very unlikely that you'll identify secondary spermatocytes. And then, of course, the spermatid phase is where the cells differentiate into mature sperm cells. These are post-meiotic cells. There's no cell division. The spermatids would be 1n1c. If we look at the spermatogonial phase, basically stem cells divide mitotically to replace themselves and to produce a population of committed spermatogonial cells. The diagram shows the stages where there's mitosis, and then the stages of meiosis, and then finally just sperm maturation. There are 3 types of spermatogonia in humans. There's type A, or dark spermatogonia. These have ovoid nuclei with intensely basophilic and finely granular chromatin. These cells divide at irregular intervals to give rise to more of themselves, or to type A, pale spermatogonia. Type A pale spermatogonia have ovoid nuclei with lightly stained and finely granular chromatin. These are the cells that are committed to a differentiation process that will ultimately produce a sperm. Type B spermatogonial cells have rounded nuclei. Chromatin are condensed into large clumps along the nuclear envelope and around the nucleolus. These cells divide by mitosis to produce primary spermatocytes. Note that type A pale spermatogonia remain attached to each other via cytoplasmic bridges. These cytoplasmic bridges remain intact throughout spermatogenesis and throughout spermiogenesis. The spermatocyte phase is where primary spermatocytes undergo meiosis to reduce both the chromosome number and the DNA content, and this diagram outlines the events of meiosis. Let's just white out the female side because we're going to just talk here about the male reproductive system. Primary spermatocytes are 2n4c. Prophase 1 lasts up to 22 days in human males. Crossing over and independent assortment, of course, assume genetic diversity of the gametes. At the conclusion of meiosis 1, secondary spermatocytes are formed. The secondary spermatocytes are 1n2c, so remember the first meiotic division is the reduction division. Second meiotic division lasts just a few hours in human males, so as I said, it'll be very difficult to identify secondary spermatocytes histologically, but at the conclusion of meiosis 2, the secondary spermatocytes become spermatids. These are the haploid cells that are 1n1c. Just remember the meiotic divisions. Meiosis 1 is considered the heterotypic or reduction division because chromosome number is actually reduced to the haploid level. Meiosis 2 is the equational division. Chromosome number essentially stays the same here as haploid, or 23 chromosomes, but DNA content is reduced from 2c to 1c. The spermatid phase is also known as spermiogenesis. Spermatids undergo extensive remodeling as they differentiate into mature sperm. We can define 4 phases in the spermatid phase. Now, differentiation occurs as spermatids remain intimately associated with Sertoli cells. The Golgi phase, there are PAS-positive granules that are known as proacrosomal granules. These granules accumulate in the Golgi. Golgi-derived vesicles coalesce into an acrosomal vesicle adjacent to the nuclear envelope, and this acrosomal vesicle defines the anterior pole of the future sperm. Centrioles migrate away from the anterior pole to initiate the assembly of the 9 peripheral microtubule doublets and the 2 central microtubules of the axoneme. This would define the posterior pole of the eventual mature sperm. The cap phase, the acrosomal vesicle spreads over the anterior half of the nucleus. The nuclear envelope beneath the acrosomal vesicle loses its pores, it becomes much thicker, and the nuclear contents become highly pycnotic as the chromosome content get highly condensed. The acrosome phase, spermatids reorient themselves so the heads are now deeply embedded in the Sertoli cells and pointing to the basal lamina, while the flagella extend into the lumen of the seminiferous tubule. In the acrosome phase, the nucleus flattens and elongates along with the overlying acrosome, moves to a position just adjacent to the anterior plasma membrane, and that gives rise to the future headpiece of the sperm. The cytoplasmic microtubules organize into a cylindrical sheath called the manchette, which serves as a guide for mitochondria to align themselves along the future middle piece of the sperm. The centrioles migrate back towards the posterior surface of the nucleus and are modified to form a connecting piece or neck region of the developing sperm. Outer dense fibers of the axoneme develop from the centrioles. As the flagellum elongates, dense fibers surround the axoneme as the principal piece. And in the maturation phase, the cytoplasmic bridges between spermatids break down and the excess cytoplasm will be phagocytized by Sertoli cells. And if one looks at sperm structure, a mature human sperm is about 60 microns long. The head is flat and pointed. It's about 4.5 microns long by about 3 microns wide by about a micron thick. The acrosome cap contains hyaluronidase, neuramidase, acid phosphatase, and acrosin, which is a trypsin-like protease. And of course, these enzymes are necessary to break down the zona pellucida of the egg. Release of the acrosomal enzymes occurs as the sperm actually touches the egg. That's called the acrosomal reaction. That's essential to penetrate the zona pellucida of the egg, and it's essential to prevent polyspermy because it causes changes in the zona pellucida of the egg. Mitochondria in the midpiece provide energy for the flagella movement. The midpiece is about 6 microns long. The principal piece is about 40 microns long. The principal piece contains the axoneme as well as a dense fiber sheath of proteins that play a role in directing the beat of the flagella. The tail piece is about 5 microns long and consists of just the axoneme. Remember that mature sperm can live for several weeks within the male excurrent duct system, but they only survive for 2 to 3 days in the female tract. And there's a process called capacitation of sperm, which occurs in the female tract. Capacitation is necessary for the sperm to gain their fertilizability of the egg. There's a morphological maturation. This occurs in the seminiferous tubules. Functional maturation occurs in the epididymis as the mature sperm gain motility. There is a certain stabilization of sperm membranes that occur in the seminiferous tubules and in the epididymis. This occurs by secretions that come from the seminal vesicle. Basically, cholesterol and other glycoproteins are added to the sperm plasma membrane. Now, as we said, capacitation occurs in the female tract, and part of the process of capacitation removes cholesterol from the sperm plasma membrane, and sugar-specific selectins, probably mannose receptors, are inserted into the sperm membrane at the equatorial region of the head of the sperm. The sperm binds to the zona pellucida. This occurs in the oviduct as sugar receptors on the sperm bind to sugar residues on glycoproteins of the zona pellucida. These are the so-called ZP1, 2, and 3 proteins. This almost certainly involves mannose-specific selectins and other mannose residues. The acrosome reaction occurs in the oviduct when the sperm makes contact with the zona pellucida. Fusion of the acrosomal and plasma membrane occurs, and the acrosome contents are released near and contacting the zona pellucida. When this occurs, the sperm moves through the zona pellucida as the acrosomal enzymes digest a hole within the zona pellucida, and of course the flagellum propels the sperm head to the oocyte membrane. Binding of the sperm to the oocyte membrane and entry of the sperm nucleus is called penetration. The sperm binds to the oocyte at the post-acrosomal membrane, and the sperm nucleus penetrates the egg plasma membrane. That initiates a cortical reaction. This is release of granules from the oocyte cytoplasm. This cortical reaction alters the glycoproteins of the zona pellucida. This is critical in preventing polyspermy. Fertilization, replication, and fusion of the pronuclei, which is called amphimixis, and the first cleavage division will occur. The male and female pronuclei decondense in their chromatin, replicate their DNA independently, then the chromosomes align in the mitotic spindle for the first cleavage division. And I've put the DNA and chromosome contents here. Basically, the male and female pronuclei are both 1N, 1C. They independently replicate DNA to become 1N, 2C for each pronucleus prior to fusion and the first cleavage division. And if you follow the diagram, I think you can see that. Just imagine the solid representation for a chromosome represents one set of chromosomes from, say, the male pronucleus. The dotted chromosomes represent, say, the chromatin from the female pronucleus. You can see they replicate DNA. They then fuse before the first zygotic division occurs. There are cycles of seminiferous epithelium, and you might want to ask how long does it take to produce sperm and what would normal sperm counts be. Because developing gametes remain joined by cytoplasmic bridges, you can see distinct cell groupings within the seminiferous tubules. In humans, there are up to 6 distinct stages or groupings of sperm development that can be observed within any given region of the seminiferous tubules, and these diagrams on the left show the different stages with some of the cell types that you might see at the different stages. I present this to you because you'll find it in the literature. You probably should just be aware that in humans there can be 6 distinct stages or groupings of sperm development within any given region of the seminiferous tubules. Now, it takes 74 days for a spermatogonial cell to complete spermatogenesis and spermiogenesis. It takes about another 16 days for sperm to pass through the epididymis. There are 300 million sperm produced daily in a healthy testis. A normal ejaculate contains perhaps 60 million sperm, and a sperm count of 10 million per milliliter of semen would represent just the barest minimal level of fertility. If you have 10 million or less, the male is usually considered infertile. The sperm count is usually considered too low. There are various factors that can affect spermatogenesis and male fertility. Some of these might be dietary deficiencies, for example, deficiencies in vitamins. There can be environmental and lifestyle issues that might affect spermatogenesis and male fertility. Some of the studies have flaws, so that's a little bit iffy to understand how environmental and lifestyle factors affect male fertility. There are, of course, developmental disorders like cryptorchism, hypospadias, and low birth weight provide significant risk factors for testicular cancer, and that, of course, can be associated with reduced semen quality. Various ciliopathies or other genetic mutations can affect fertility. A notable one is Cartaginer syndrome, where the axoneme doesn't function. You can have systemic diseases or local infections of the testis, perhaps fever, kidney disease, HIV, maybe other viral infections or metabolic disorders might affect sperm production. Elevated testicular temperature is an interesting one. The main reason the testes have to descend is because sperm maturation and functional beating of the flagella only occurs if the sperm are a few degrees lower than body temperature. Of course, medications might affect fertility. Exposure to synthetic estrogens or other sex steroids might exert a negative feedback on FSH secretion. Prenatal exposure to estrogens might inhibit Sertoli cells. It even turns out that some drugs that are used to treat enlarged prostate can have a negative effect on fertility. Toxic agents, mutagenic agents might affect fertility. Ionizing radiation or other alkylating agents can affect rapidly dividing cells, and hence a spermatogonia could be affected.